Good afternoon. Today's lecture deals specifically with damage of DNA and repair of DNA. Our last series of lectures focused on the mechanisms of gene expression, particularly how information flows from the genetic material to a final functional phenotype that we can assess. Those lectures focused on replication of the genetic material, transcription of the genetic material, and translation of RNAs that were the result of transcription of protein-coding genes. And at the tail end of that last lecture, we touched on the concept of mutations and what effect a mutation at the nucleotide level could potentially have on the gene product downstream. Now, what I want to do a little bit today before we actually get into the heart of the material is define what a mutation is. A mutation is not necessarily something that is bad. It does have a negative connotation typically associated with it, but the definition of a mutation is a process that produces an alteration in DNA sequence or chromosome structure. Now, in certain circumstances and in many circumstances, this can in fact be detrimental, but it is not always detrimental, like what we talked about at the tail end of the last lecture. So again, a mutation is a process that produces an alteration in DNA sequence, typically when you compare that to a reference sequence, or an alteration in chromosome structure. For example, several lectures back, we talked about modification of chromosome structure, such as changes in the number of chromosomes, deletion of chromosomes, duplication, and rearrangements. However, today's lecture is going to focus on mutations at the DNA sequence level. Now, mutations can be classified in lots of different ways. The first way I want to discuss is the classification of mutations as being something that's referred to as spontaneous versus induced. I'll start with induced mutation. An induced mutation is a mutation that results from the influence of an artificial factor. So there is some component, something artificial, that is causing the change in sequence or the chromosomal damage. That is different from a spontaneous mutation, which is defined as naturally occurring and is not induced by any type of specific artificial factor that would be referred to as a mutagenic agent. So again, two broad categories as far as classification is concerned is how the mutation arises. Is it induced by an artificial factor or does it just happen? Typically, the just happens is situations where mistakes take place, and we will get to some of those examples in just a little bit. Another way that mutations can be classified is whether they are gametic mutations versus somatic mutations. Now, a somatic mutation is one that occurs in a somatic cell, and this is usually masked in the individual. Somatic cells are cells that are different or distinct from gametes, and gametes are the sex cells that allow for the transmission of genetic information to the next generation. So if a mutation occurs in a somatic cell, that is not going to be typically passed on to the next generation. It will only have the effects in the individual that the mutation occurs in. Something a lot more serious might be something that's referred to as a gametic mutation. A gametic mutation occurs in the genes or chromosomes found within the gametes or gamete-forming tissue. This then has the potential of being passed on from one generation to the next. So if you look at the slide with point number 1, we have a somatic mutation occurring in a non-reproductive type of cell in this particular fish. That somatic mutation can be amplified through mitosis as the organism continues to grow, and you have more cells that have that particular mutation. So in this case, you have a population of mutant cells, but in the long run will only potentially cause problem to that individual and will not be passed on to the next generation. Along the bottom of the slide, you see the alternative example where you may have a mutation that occurs in germline tissue, in gametes or gamete-forming tissues. In this particular case, that mutation would be passed on to the next generation, and those mutations then are carried on to other individuals that result from this particular individual. And so this might be something a lot more serious because of the longevity and passing on from one generation to the next. So the first two slides dealt specifically with categorization. These types of mutations were exactly what we discussed at the tail end of the last lecture. Base substitutions, like you see in this table, result in a change for one nucleotide for another. We discussed also at the tail end of last lecture that those can have different effects. A substitution can result in what's known as a silent mutation, and that silent mutation results in no change in amino acid because of the redundancy of the genetic code. Substitutions can also fall into the class or category of being a missense mutation. And in a missense mutation, this substitution results in a different amino acid being inserted than what was originally coded for. So the single nucleotide change results in a different amino acid being inserted during the protein synthesis process. And lastly, a substitution can be categorized as a nonsense mutation. And a nonsense mutation is where the codon has been altered to become a stop codon. This is considered nonsense because what happens during protein synthesis is a premature termination of the protein synthesis process because the original codon has now been changed to a stop codon and protein synthesis stops. Now, insertions or deletions can actually be much more detrimental when we are talking about the downstream effects as far as protein synthesis is concerned. The insertion of a nucleotide or the deletion of one or more nucleotides will then alter the reading frame in the protein synthesis process. If you recall, protein synthesis again starts at the AUG codon, and this sets up what's known as the reading frame. From that point on, every 3 nucleotides is a codon and codes for the insertion of a specific amino acid. If you have now inserted an additional nucleotide or deleted a nucleotide, the entire reading frame has been shifted, and what results is a protein that is essentially gobbledygook, doesn't make sense, and oftentimes results in premature termination. A third way that point mutations can occur is what's referred to as expanding nucleotide repeats. There are certain genes that have codons that are repeated multiple times within the gene in their normal circumstance. So if you look at this table, we have a number of different disorders that are listed here where the number of repeats of a given codon is altered from the normal range. So let's look at the first example, which is spinal and bulbar muscular atrophy. So the repeat sequence is CAG, and in the normal copy or the normal allele, there are somewhere between 11 to 33 repeats of CAG. In individuals with this particular type of muscular atrophy, there is a dramatic increase in the number of these particular repeats, anywhere from 40 to 62. And you can look through this table and see multiple examples of that overall. This typically occurs with problems due to replication errors. The best analogy that I can give for how this happens is that if you think about reading a sentence, us as individuals reading a sentence, and if the sentence had the word the, a 3-letter word, repeated over and over and over again, as you're reading that sentence with the word the, you might lose your spot and accidentally start all over again. And this is exactly what happens during the replication process, is the enzymes sort of lose their place in the process and begin to add in additional repeats than what are normally called for. So also at the tail end of the last lecture, we looked at the specific example of point mutations with respect to the disease or disorder known as sickle cell anemia. That is a classic example that is utilized because it is a single nucleotide mutation that results in a single amino acid substitution that then results in a misshapen hemoglobin protein, hence the misshapen red blood cells in the individuals afflicted with this particular disease. So that is something that might be detrimental in that mutation example. However, if you look at the slide here, the human ABO blood types, or the multiple types of blood that we know exist, are due to nucleotide mutations. Now, hopefully you are all familiar with the concept that there are, in fact, 4 different blood types in humans. Humans can have type A blood, type B blood, type AB blood, and type O. Now, we discussed this way back at the beginning of the semester when we discussed multiple alleles, as well as codominance and incomplete dominance. And if you recall, what we discussed was that individuals with type A blood had the A form of a specific protein associated with their red blood cells. Individuals with B blood had the B form of this particular protein on the surface of their red blood cells. Individuals with type AB blood have both the A and the B form of the protein on the surface of their red blood cells, and individuals that are type O blood don't have this particular protein on the surface of their red blood cells. What you then see at the bottom half of this slide is an example of the enzymatic pathway that results in the production of the proteins that are in question here, the type A or type B protein. So in this biochemical pathway, there is something known as the H substance. There is then an enzyme known as a glycosyltransferase. And in the first example, this enzyme allows for the production of the A form of the protein. There is a distinct glycosyltransferase that then allows for the production of the B form, and there are 4 nucleotide differences in the genes between these 2 enzymatic gene components. So here you can see base substitution does result in different protein products in the end. More dramatically, if you look at the very last example for what happens in type O blood, is that this particular gene has been mutated with a frame shift. And with the frame shift, this results in a non-functional protein, specifically the glycosyltransferase, hence the X over the arrow, meaning this enzyme is not functional. If the enzyme is not functional, then the conversion of the H substance doesn't occur, and hence these individuals do not have this protein on the surface of their red blood cell. So again, we see multiple examples of what can happen with nucleotide mutations with the ABO blood type. Simple base substitutions result in subtly different enzymes that get produced, which then results in different proteins being produced and found on the surface of the red blood cell. Also, these frame shifts, which is either an insertion or a deletion, results in a non-functional protein product, hence the enzyme is not present, doesn't work. These proteins are not found on the surface of the red blood cell. Okay, so now let's transition a little bit into what actually could be causing these mutations that we just discussed in the previous few slides. One example of how a mutation can in fact occur is what's known as a tautomeric shift. A tautomeric shift is a mutation that results from a shift in the localization of a hydrogen atom. Essentially what that means is that isomers can form with specific nucleotides. So there can be isomer formation with specific nucleotides. And when these isomers do in fact form, it allows for an incorrect type of base pairing to take place. So let's look at the examples that we see in this slide or this figure. In panel A, you're looking at the standard base pairing arrangement that we know exists. The base pairing rule, the sequence complementarity that we've talked about multiple times throughout the several past lectures. Thymine, always hydrogen bonding with adenine, and specifically they fit together like puzzle pieces with 2 hydrogen bonds allowing for that interaction. On the right-hand side in panel A, you see cytosine, hydrogen bonding with guanine. Again, this is our normal, typical base pairing that takes place with 3 hydrogen bonds that fit together like puzzle pieces to allow cytosine and guanine to interact. However, in panel B, what you can see is these tautomeric shifts. They are a shift in the localization of a hydrogen atom, resulting in an isomer to form. So what you see with thymine is that this isomer now allows for 3 hydrogen bonds to form instead of 2, and specifically that allows for base pairing with guanine. You can see how this could be a potential problem during DNA replication. If the template sequence has a T, then the newly synthesized DNA should get an A as the complementary sequence. However, if that T has undergone a tautomeric shift, now the replication machinery allows for the base pairing of T and G, and now we've inserted an incorrect nucleotide during the replication process. And this is how a base substitution can occur, specifically in this category of a tautomeric shift. And a similar example can be seen on the right with cytosine undergoing the tautomeric shift, allowing now for 2 hydrogen bonds to form, which then allows it to base pair with adenine rather than guanine. Another cause of mutations, specifically insertions and deletions, is due to what's referred to as mispairing. This too is a problem that occurs during DNA replication, specifically when there is a stretch of nucleotides that are exactly the same. So in this example at the very top of the slide, you see the template strand, and it has a stretch of T's, one right after the other, right smack dab in the middle of that sequence. On the top, you see the newly synthesized strand, which would then have a stretch of A's because of the sequence complementarity. However, there could be an error with that newly synthesized sequence where a slight loop occurs where one of the A's doesn't base pair anymore or is mismatched. And you can see that little blip in portion or panel 1 of this figure. If that does in fact occur, if you follow that through the replication process, there is this blip that remains in the sequence overall. This would then result in the addition of a nucleotide in the newly synthesized strand. Alternatively, if you look at panels 3 and 4, you could have the looping out occur on the template strand rather than on the newly synthesized strand. And again, these are all mistakes or errors that could in fact happen. Don't necessarily always happen, but could happen simply by those hydrogen bonds releasing and falling back in the incorrect position. If you follow that through to panel 4, you would see that there is a omission of a nucleotide, a deletion of a nucleotide that has occurred in the newly synthesized strand. So in either one of these situations, there is a problem that has occurred, either the insertion of an additional nucleotide or the deletion of a nucleotide. Another cause of mutations would be the insertion of a base analog. A base analog is a molecule that can be substituted for either a purine or a pyrimidine during the nucleic acid synthesis process. What happens if this particular analog gets inserted is you now have a nucleotide that has a base that looks very similar to one of the quote-unquote normal bases and could potentially be inserted during the replication process. The example that you see here is a analog known as 5-bromouracil. 5-bromouracil gets mistaken for the base thymine and can in fact get incorporated into a nucleotide. If it is incorporated into a nucleotide, when replication occurs, what you can see on the right-hand side of this figure is that what would base pair now with 5-bromouracil is guanine. So again, 5-bromouracil is an analog for thymine. It structurally is very similar to thymine, so it would be inserted as a thymine would. However, when replication takes place, you can see that you have the incorrect type of base pairing that would now occur. And this is just one specific example of a base analog. There are other examples of molecules that again are structurally very similar to other bases and can get mistakenly incorporated into a nucleotide. And if that happens, that nucleotide with an incorrect base could be incorporated into the DNA, hence the mispairing that might occur during DNA replication. Another way that mutations can in fact occur is at the time of crossing over or genetic recombination. If homologous chromosomes misalign, that means they don't align the way they're supposed to during the crossover or genetic recombination process, you can have something that's referred to as unequal crossing over. And with unequal crossing over, you have resulting chromosome structure that has different genetic information in it than it did previously. And so here too, you can have insertion or deletion of stretches of nucleotides with mistakes occurring during the crossover or genetic recombination process. Other ways that point mutations can in fact take place. The next example is a category of causes that are due to chemical mutations. Chemical mutagens. Chemical mutagens are chemicals that can cause DNA damage. They change the DNA in some way, and that can result in detrimental downstream effects overall. What you see here is an example of several different mutagens and what exactly is happening when that mutagen is damaging or altering the structure of the DNA. What you see in this example in panel A is guanine as the original base. If it is treated with a specific mutagen known as EMS, you end up with alkylation, which changes the structure of guanine. With that structural change, again, you have a mispairing event that will take place. Guanine normally hydrogen bonds with cytosine. However, if it has been mutated with this alkylating agent, it will now hydrogen bond with thymine. And you can see the other examples in this particular slide as well. So in the last slide, you saw chemical mutagens that can cause changes in base structure, which eventually results in mispairing events that can take place. There are other chemical mutagens that physically cause damage to the DNA. These are things that result in destruction overall of the structural integrity of the DNA molecule. What you see on the slide are a number of molecules that actually can bind and get in between the bases in the DNA structure, like you see in the right-hand panel. And when it does that, it actually causes a distortion of the helical shape. This makes a very unstable type of DNA molecule and also a molecule that is very hard to replicate. The last two examples of causes of DNA mutations are related to one another in that they both cause structural damage of the DNA. The first example, labeled number 7, is UV light. Ultraviolet light does in fact have a great deal of energy associated with it. In particular, if there is a DNA sequence that has two thymines, one right next to each other, the exposure to UV light can cause what's known as a thymine-thymine dimer. And if you look at this figure, you can see that what happens is structurally there is covalent bonds that have now formed between these neighboring thymines. This then causes a structural distortion in the DNA. And then lastly, what you see as number 8 is ionizing radiation. These would be things like X-rays or gamma rays. These typically destroy the connection between the nucleotides, like you see labeled here with a phosphodiester bond. And with the breakage of these bonds, this then causes, again, physical damage in the DNA. As you can imagine, it turns out that there are multiple repair mechanisms that have evolved in a way to help fix some of the problems that we've just discussed. Now, not every single problem is found and fixed, but there are, again, plenty of repair mechanisms that are evolved in such a way to help fix these problems. We are going to touch on a few of those. The very first one is a mechanism that is known as mismatch repair, also abbreviated MMR, and this occurs in both prokaryotes and eukaryotes. Just to give you an overall idea of what happens in this, and then we'll get to some specifics in a minute, is that there are enzymes involved that scan the genome for mismatches. Mismatches are where incorrect nucleotides have been added in, finds those mistakes, and then fixes them. Okay, so let's talk a little bit about how that happens. There is an enzyme known as DAM methylase, DAM methylase. This is an enzyme that adds methyl groups to adenines prior to replication. This occurs prior to replication, like you see on the left-hand panel. It basically acts as a marker to label which strand was the original parent strand because of these tags or methyl groups that you can see as the little red methyl groups associated with the DNA. So again, this tags the DNA in such a way so that the enzymes involved in the repair have a reference to know which strand was the original parental strand and which strand is the newly synthesized strand. Again, the newly synthesized strand will not have any of these methyl groups added to it. In the same left-hand panel, panel B, you can see an enzyme known as MUT-H. MUT-H is an enzyme that recognizes these sites that have a methyl group on one strand and no methyl group on the other strand. Again, this enzyme helps with the recognition to know which strand was the original template strand and which strand was the newly synthesized strand. However, this enzyme is basically no good alone. It just helps with the recognition of which strand was the original versus the newly synthesized. In the right-hand panel, you see an additional enzyme known as MUT-S. MUT-S is a molecule that occurs in 2 copies, hence it's referred to as a dimer, and it is a functional repair protein. It has this characteristic horseshoe shape, and it scans the DNA to find where MUT-H has located a tag for methylation. In this slide, we can take a little closer look at what's happening with MUT-S. MUT-S scans the DNA for mismatches. Mismatches would be where there's physical distortions that have occurred because an incorrect nucleotide has been added in. The mismatch is recognized, and additional enzymes known as MUT-L and the original MUT-H that I mentioned in the last slide are recruited to the target site. These are accessory proteins in this repair process. In particular, MUT-H is an enzyme known as an endonuclease. It is a specific type of enzyme that can make cuts in the DNA. Additional enzymes are recruited to the target location, including a helicase known as UVRD, as well as an additional exonuclease to help cut out damaged or mismatched DNA. The result that occurs with the recruitment of these enzymes is that a single strand gap has been generated, and hopefully you can see that on the right-hand panel where a stretch of DNA has been removed by the enzymes in question. These enzymes now are removed, and there is a gap that DNA polymerase III can come in and fill, and ligase will close in that last covalent phosphodiester bond. So essentially what's happening here in this mechanism, to summarize it, is a mismatch is recognized, then a piece of the newly synthesized DNA is cut out where the mismatch occurs. The gap that's left behind is filled in with new DNA, and hopefully this time no mismatch will take place. This next repair mechanism that I want to highlight is what's referred to as direct reversal of DNA damage. This is also a mechanism that occurs universally, meaning it's found in both prokaryotes and eukaryotes. What happens in this mechanism? Actually, let's take a step back. This particular mechanism is known as photoreactivation, and it involves fixing thymine-thymine dimers that may have been formed due to UV light exposure. So a few slides back, we discussed the fact that UV light can in fact cause these thymine-thymine dimers to form, where covalent linkages occur between neighboring thymines. This causes a physical distortion in the DNA, and that is recognized by an enzyme known as DNA photolyase, and you can see that in the left-hand panel. This enzyme recognizes the distortion, and it uses the energy from visible light to break those covalent bonds. Hence, the thymine-thymine dimer is removed in the sense that there's no more dimer, there's no more covalent bond between the neighboring thymines, and the DNA is no longer distorted. The last two examples are types of excision repair, but they are subtly different from one another. So the example that you see on this particular slide is known as base excision repair, also known as BER. In this particular example, there are additional enzymes that are involved in this particular repair process. There are enzymes known as DNA glycosylases. These are enzymes that recognize incorrect bases and remove by hydrolyzing the glycosidic bond. Essentially, what happens if you follow the sequence of steps in this particular figure through, what you can see in this example is that for some reason there is a base that contains a U in this double-stranded DNA molecule. Now, U's are not supposed to be found in DNA, hence this is recognized as a problem by one of these specific DNA glycosylases. The problem is recognized, and specifically the base itself is cut out. So not the entire nucleotide, but just the base. Once the base has been removed, this is now recognized by other enzymes as a physical distortion, because now there is no base that allows for hydrogen bonding between the two strands at that particular point. There is an enzyme known as AP endonuclease that comes in and makes a cut now to remove the remaining portion of the nucleotide. Now there is an actual gap in the sequence where an entire nucleotide is missing. This is recognized by DNA polymerase, typically DNA polymerase I if we're talking about prokaryotes, fills in the gap, and then ligase closes that covalent linkage that needs to be closed. This last example is also a type of excision repair. However, this is referred to as nucleotide excision repair. In this case, damaged DNA is recognized by enzymes that are part of the UVR gene complex. They are UVR gene products. With this specific example, we're noting a thymine-thymine dimer, and that is recognized by a number of enzymes in the process. The enzymes recognize the distortion and make a cut on either side of the problem. So as you can see where the thymine-thymine dimer is formed right at the center of the slide, there's a cut made on either side, and that piece that has the thymine-thymine dimer is removed. Now there is a gap in DNA sequence that can then be filled in with DNA polymerase I and the final covalent linkage sealed up with DNA ligase. Again, there are multiple examples of repair mechanisms. What I wanted to do was highlight just a few for you so that you get a feel for the fact that there are plenty of mechanisms that have evolved to help fix DNA damage. Again, this is just a small snippet